Hello, hello. Welcome to AP Biology Review, and we have many things we have to go over for Unit 3. We've had 2 so far, obviously. I haven't made anything on them yet, so I'll get to that. Alright, so we have metabolism. Metabolism is all of the chemical reactions that happen, so digestion, and all of that stuff. And there's 2 types of series of reactions called metabolic pathways. We have catabolic pathways, which release energy by breaking things down, just like that, and then anabolic pathways, which create energy by combining together. And we have laws of thermodynamics, which are about how heat works and stuff like that. So, rule number 1, energy cannot be created or destroyed. You've probably learned this one very early on, so you should know that one quite well, because it can only be converted into other forms of energy, so chemical energy into, say, light energy and stuff like that. Second law, when energy transforms, as I mentioned before, it usually becomes unusable in some way, and some of it is lost as heat, and so slowly more chaos or entropy is created in the universe. And as you saw before, we had catabolism, anabolism. Here's a little diagram. So catabolism, we have the molecules breaking down the energy, and anabolism, they're combining, taking in all that energy to become one whole. And let's measure free energy, which is another way to measure energy, which is how spontaneous reactions are. And we have this beautiful, terrifying formula, which will certainly traumatize you. So we have the delta G, which is the change in free energy, so the change in how spontaneous it is. And we have total energy, so not just how spontaneous it is, but everything else. Then entropy specifically, which is that heat we release, and temperature, which is average kinetic energy. So, total energy minus the amount of energy that's being released as heat and the temperature, and you get free energy. Alright, and measuring free energy, we have 2 reactions. Yay! So, exergonic energy reactions, which are the ones that release energy. So, we go from being less spontaneous to becoming more spontaneous, such as cellular respiration. And then we have endergonic reactions, not like the Ender Dragon, which become less spontaneous over time, such as photosynthesis. There we are. And next up, we have an important thing to ask, which is, what is ATP? So ATP is this beautiful molecule. It's similar to a nucleic acid, as in it has adenine, which is the A type on the DNA, and a ribose sugar. And then it has 3 phosphate groups. And when those phosphate groups are broken, the second and the third one specifically, ATP releases energy and becomes ADP, adenine diphosphate instead of triphosphate. And this process is known as phosphorylation. Alright, that's basically the basics of reactions, so we're going to go into enzymes. So enzymes are proteins that speed up reactions by lowering the energy required for them to occur. This is shown over here, because you need some sort of energy to occur of it going into a less spontaneous state for it to become more spontaneous and more reactive. And here is the basically biology of an enzyme. We have substrates, which are these particles that go into the enzyme through the active site. They bind there, and They do this through, hold on, induced fit right here. So they kind of go in and then the enzyme clamps around them. And as they are proteins, their temperature can be affected by, their structure can be affected by temperature, pH, and other chemicals. So if there's a really high or low pH, the enzyme will not be able to hydrogen bond and it won't be able to fit together anymore, so those reactions won't be able to occur. And for the temperature, it's just going to become like scrambled. It's going to become DNA. So when do enzymes work the best? Well, temperature and pH that allow them to function optimally. So usually for temperature, as I said, I did not say actually, I'm a liar. Oh no. So temperature, the activity goes up because more collisions are occurring, and then after that it goes down because the enzyme denatures. And for pH, it's really dependent. It depends on which pH it works best. But again, hydrogen bonds will break it. Okay, so we have enzyme inhibitors, which stop certain enzymes from working, and there are two types. Permanent, which permanently stop it. You can't remove them, and then the enzymes will no longer function, your reactions will slow down, and then you die. Maybe. Hopefully not. And then we have reversible, which can be removed. And we have two types of these two types. We have competitive, which, such as this circle, will bind right here and stop the substrate from entering. And then we also have non-competitive, which instead bind down here, changing the shape of the enzyme so that the substrates cannot enter instead. And this place is known as the allosteric site, which is the part that changes the enzyme's shape. There are several other ways that they can change the shape of the enzyme. They can actually make it become from inactive to active by binding to that site and then opening up the little enzyme active site. Or they could instead bind to the active site and to open it all the other parts of the enzyme that way as well. Alright, and that is enzymes. Oh, and now we have photosynthesis, my second least favorite part. Sure, no one likes it, learning about it. It's like, anyway, back to, now we have photosynthesis. So, first we have to know the difference between autotrophs, heterotrophs. Probably learned this already. So, autotroph, they produce their own food. Heterotroph, live off of others. Also cute bunny. Anyway. So yeah, now how did autotrophs become autotrophs? Well, they came from cyanobacteria, which were the earliest capable organisms that could do photosynthesis, and they actually made the oxygen so that the heterotrophs, bunny, could live. Also a fun picture. Alright, now, where does photosynthesis happen? So, again, if you paid attention in Unit 2, you know that photosynthesis happens in the chloroplast. And the chloroplasts, which you may not know, are found in the mesophyll, which is the interior tissue of the leaf. And they also have stomata, which are like these little holes right here to control the carbon dioxide, water, and oxygen entering. More about that later. Alright, here's the simplified version. We're about to get into the nitty-gritty. So, carbon dioxide, water, and light create sugar and 6 oxygen. So, here's that in more detail with specific numbers. And there are 2 stages to photosynthesis. We have the light reactions and the Calvin cycle. And another thing to remember before we get into the light reactions and the Calvin cycle are the redox reactions, which is when electrons are moved from one thing to another. So Leo goes to Ger, oxidation equals losing electrons, reduction gaining electrons. And when you gain electrons, it's like a payload. You become more sad, more negative. You move more slowly. And another thing to remember is that in order to absorb light, there are photosynthetic pigments. So these pigments absorb visible light, and they reflect the things that they do not absorb. So that's the colors we see. So we have chlorophyll A, which is the primary ones used in plants. It's blue and green. And we have chlorophyll B, which is the yellow-green pigment, and together they work to absorb the necessary wavelengths for the light reactions. And then we have carotenoids, which are the yellow and orange ones that absorb extra light that is unnecessary, and the plants would like to get rid of because too much light, such as like too much radiation and stuff, is also dangerous to the plants. And here is some terrifying chart that we will about to get into. So, hold on to your horses. Another thing to remember before we get to the traumatizing chart. Here, so you don't have to look at it too much. Alright, there are NADPH and ATP molecules. These hold the light energy from the photons, and they have excited electrons in them. So, let's begin. First, we have chlorophyll. Let's put this down here for a reference. There. Alright, right in these two proteins, which we'll get into. The chlorophyll boosts the electron into an excited sort of state, and it emits more photons in fluorescence. Now, where does this happen? I told you in proteins. So the photosystems, which are where the reactions occur and the light is captured. So there's a reaction center. What's the reaction center? It's lots of proteins with chlorophyll A and an electron acceptor, which takes in finally that last photon bouncing around and then sends it off. And of course we have the pigments as well, so which absorb all that light that bounces around inside. So here we go. Photosystem 2, the first one. Where's my picture? Hold on. There we are, just so you know. Photosystem 2 is right here. So, you absorb the light. It bounces around from the electrons. It goes into the primary acceptor, which is then sent into this protein right here, plastoquinone, then cytochrome, then PC, and then photosystem 1, which we'll talk to in a moment. Meanwhile, in order to replace the electron that bounced away, water is split, and they rob its electrons, 2 electrons from it, into the protein. And then there's 2 hydrogen ions produced over here, keep them in mind, and 1 oxygen molecule, which will then combine to make O2, and that's just released. That's one of the 6 O2 molecules produced in photosynthesis. So, photosystem 1, which we've traveled to, more light comes in and excites another electron, see light coming in, which then goes to ferredoxin and then NADP reductase. So, remember those hydrogen ions that are all the way over here? So, first of all, they're coming all the way through ATP synthase right here. ADP is combining to make ATP, and that's going to be used in the next part, which is the Calvin cycle. And in the meantime, those electrons are combining with the hydrogen ions over here to turn NAD+, which is the version without the electrons, into NADPH, which is the reduced version because it gained electrons. And then NADPH and ATP are about to be used up in something awesome. And before we get into that awesome thing, remember, specifically, photosystem 2 absorbs 680 nanometer light, photosystem 1 absorbs 700 nanometer light. And also, they're in reverse order for some reason, because they want to torture you. Alright. So, the Calvin cycle. We have 3 parts: carbon fixation, reduction and regeneration of rhubarb. Weird name. So, carbon fixation is when CO2 is incorporated into the Calvin cycle one at a time. Let's get a picture, because I love pictures. Here we are. A little scary picture, but we'll get to it. So, here, carbon dioxide is entering the cycle, and it's attaching to rhubarb right here, this molecule. So together they combine and they create 3-PGA, or 3-phosphoglycerate, which is this beautiful molecule. Then we have reduction, where ATP is giving away its electrons, becoming ADP. Rather, it's phosphorylating it and becoming ADP. And then NADPH is giving it electrons. That reduces it, and so it becomes 1,3-bisphosphoglycerate first, and then it becomes G3P, which one of those G3P will be sent away. Here, hold on. Reduction. Here we are. Anyway, one of those three molecules becomes used up, because remember, we're adding 3 CO2 in total, which creates 6 G3P. One of those G3P will be used up for real to make sugar. We're not really going to get into that process, so just remember that's the sugar that is made in photosynthesis. And then the rest of it's going to go up here, and then we're going to get to phase 3, which is regeneration of rhubarb, because remember, this is a cycle. So we take the rest of the G3P, add more ATP in to phosphorylate it, turns back into ADP, and we're back in with rhubarb to add more CO2, and then repeat this cycle, and boom, we get lots and lots of G3P, which then becomes sugar. I need to stop doing that. So, little quick recap. Boom. So, the Calvin cycle specifically, we add in 3 CO2, 9 ATP, which was made in the light reactions, and 6 NADPH, which was made from there, and make 1 G3P molecule, which turns into sugar, 9 ADP, and 6 NADP+, which will then be used up in the light reactions to create ATP and NADPH. It's raining. All right. So, adaptations made by plants because of photorespiration. Remember the stomata that open and close? Why do they do this? To stop water loss, because the water is going to otherwise evaporate, so they close. But this causes there to be less carbon dioxide, which, remember, is necessary in the Calvin cycle. And so, this causes rubisco to bind to oxygen. And by the way, rubisco is the enzyme used to create combined RUBP and CO2. And so that binds to oxygen and uses up ATP. So no sugar is produced. It's bad for the plant, even though the process is producing CO2. It's way less efficient. So, some adaptations. We have C4 plants. Those were C3 plants, by the way. So, they separate their steps, so they have one mesophyll cell, which takes in all that CO2, combines it into a 4-carbon, and then that's transferred to a bundle sheath cell, which actually does all of the Calvin cycle stuff. So this allows the stomata to be partially open to slowly absorb a little bit of it, which can then be stored into the 4-carbon molecule in the mesophyll cells, and then be used up. Then we also have cam plants, like a camera, I guess. I don't know. They named it for that. Who knows? So, these are open their stomata in the night when less water is likely to evaporate to absorb all that carbon. Then that is incorporated, added into organic acids. And then during the day, that is going to be used in carbon dioxide and incorporated into the Calvin cycle. So, basically, cam plants open in the night, closed in the day, and they do this all in one cell, and remember, the C4 plants do it in 2 cells. And C3 plants suck. So that's photosynthesis. And moving on to cellular respiration. Right. Welcome to part 3. We've had an increase in budget, as you can see by this 1 penny right here, minted in 2016. And we're going to spend it. Oh. And now we have full color. Beautiful. So, cellular respiration, which is where we're using up that glucose. We have it right here. chemical energy in glucose, organic molecules. There's also fructose, sucrose, other stuff. They use it to make ATP. Lots and lots of ATP. Remember, light reactions only made 9 or 6 ATP, and this is about to make 30, so leaps and bounds ahead. So, we're going to break it down, glucose, and remember, we're going to be using those redox reactions. So, especially here where the sugar is going to be oxidized into carbon dioxide, and the oxygen will be reduced into water. And here's our general breakdown. So we have sugar, oxygen equals carbon dioxide, water, and energy, which is the ATP we want. And this is actually, you breathe in oxygen, right? And then you release carbon dioxide and water. Alright, so let's go. Whoop. Here are the 3 steps we need to know. Glycolysis, pyruvate oxidation, and the citric acid cycle. Citric acid is actually lemon, so technically the human body runs on lemonade. Good to know. And the electron transport chain. Let's put this up here. So here, let's go. Glycolysis. Glycolysis is when you take in glucose, you create a little bit of ATP, just 2, and you create a little bit of NADH and water. But what you really care about here is that pyruvate that you make. And this looks simple. Okay, so this looks scary. Yes, I know. So what's happening? We take glucose. ATP gets turned into ADP. Some more reactions, more ATPs turn into ADP. So we're losing energy here. Then, up here, we have some NAD pluses turned into NADHs. By the way, not NADPH, different molecule. Then right here, 2 ADP turned into ATP. So we've got back all the ATP we lost. And then we create 2 more right here. So we got 2 extras left. And we have the pyruvate we make. So, glycolysis. But really, you need to know this one. This one's the simpler one. And, of course, here's some more stuff to know. It's in the cytosol, so outside the mitochondria, just floating around. And it splits glucose into 2 pyruvates. 6 carbons in glucose, 3 carbons in pyruvate. Then we have pyruvate oxidation, which is quite simple. Pyruvates just turned into acetyl-CoA, a new molecule to be used in the lemonade cycle. I mean the citric acid cycle. And that's it. That's basically all pyruvate oxidation is. So citric acid cycle. Lemons. Also known as the Krebs cycle, if you like that name better, but I like lemons more. So, all right. So, what is the citric acid cycle? Well, it enters the mitochondria, as you can see here. Bloop. It's already in the mitochondria. Pyruvate oxidation was already there. And, of course, it's oxidized to make citrate. Here's a little picture of the citric acid cycle. More wheels. Scary, scary wheels. So, let's go through it. We have pyruvate becomes acetyl-CoA. So it releases a little bit of carbon dioxide as well. Forgot to mention that. Citrate becomes different molecules and goes back to this molecule right here, combines with acetyl-CoA and restarts the cycle. What does the cycle make? Well, this cycle creates lots of delicious molecules right here in the simplified wheel. You put into acetyl-CoA, you get 2 ATP, a little more energy, 6 NADHs, 4 carbon dioxide, which is just waste, and 2 FADHs. Now you really need to focus on the NADH and FADH because that's going to be useful for the electron transport chain. And here's some more notes in case you're interested. Got that? Good. I don't care. I'm kidding. Please don't demonetize me. All right. So we have the electron transport chain right here, which is in the inner membrane of the mitochondria. So As you can see here, we got the outer membrane, and then the inner membrane is right here separating the orange and the red inside. And it's a ton of proteins, just like the light reactions. So let's go through it. And then we get a little recap, and then you can go home. I mean, you're probably at home. I don't know. So, NADH brings its electron. FADH brings its electron. They all travel through. And this traveling of electrons allows these hydrogen ions, also coming from the NADHs and FADH2, to travel through, be pumped through, all over here. And this huge concentration of them decides to go through ATP synthase in a massive flood, and that turns ADP into ATP and produces 30 or 32 ATP molecules, which is a lot more than, say, glycolysis, which just makes 2. So then these hydrogen ions combine with the electrons and the oxygen. The electrons go with the oxygen together, and the hydrogen ions then create regular old water. And then you're done. You made the ATP. So let's see it all summarized. Glycolysis takes in 1 glucose, but creates 2 pyruvate. Remember, glucose has 6 carbon, it makes 3, so it's getting cut up. 2 ATP, which sometimes is good enough for some bacteria, and 2 NADHs, which are going to be used. Alright, next you take the 2 pyruvate and pyruvate oxidation, turn it into acetyl-CoA, carbon dioxide, and NADH. You really need to know about the acetyl-CoA because it's used in the lemon cycle to create carbon dioxide, which again is wasted, don't need to worry about that. ATP, which is just there for fun as a bonus. But the NADH and FADH are important because then they donate the electrons and protons to create ATP in the electron transport chain, also known as oxidative phosphorylation and chemiosmosis. Chemiosmosis is actually more of the part over here of the flow of electrons, and the electron transport chain is more of this. And altogether, it's oxidative phosphorylation. And together, with all of the powers combined, you get 30 or 32 ATP. But this number varies. It's not fully accurate. And now we have one more issue to solve, which is what if you don't have oxygen? Then you have anaerobic respiration, which makes ATP even though there is absolutely no oxygen to use from. Why is that bad? Because they need the oxygen to absorb all the electrons in the end, and they don't want electrons floating around everywhere, because that's not beneficial. So we have 2 options. We have lactic acid fermentation, which is right here. So we use glycolysis. Remember, glycolysis makes 2 ATP over here, but then you're left with this pyruvate and NADHs, and you can't really do this process again. So you convert the pyruvate into lactate. Whoopsie daisy. And then that lactate goes off. And this process also takes those NADHs, turns them back into NAD pluses, so they can be used again. And glycolysis can be used. Does not produce as much energy, though, so that's unfortunate. And we used to be told that this causes muscle soreness, but that's a lie. That's not true at all. So don't trust that anymore. But it does produce lactate, which is a little annoying as a byproduct. There's also another thing which we do not do which is fermentation, because fermentation is making wine, and humans do not make wine. So, do I have a yes? Nope. So, fermentation is a very similar process to lactic acid creation, but instead of, as you see here, where it turns into lactate, it actually becomes alcohol instead, and this also produces carbon dioxide as a byproduct. So that's the alternative bacteria have, and this is used if you're making bread with yeast, where it produces carbon dioxide, and that causes the rise, the delicious bite of bread. Nom nom. And yes, here we are with a final little recap, really quick, and then you can be done. So we have glycolysis. If there is oxygen, then we have pyruvate oxidation, which, remember, glycolysis makes pyruvate, that makes acetyl-CoA, used in the citric acid cycle to make those electron-carrying molecules, and then that's used in the electron transport chain to make loads and loads of ATP, 30 or 32. If there is no oxygen, then glycolysis will instead do fermentation to create lactic acid or alcohol, and that produces 2 ATP each. And that is Unit 3. We have photosynthesis, enzymes and metabolic processes, and of course, the cellular respiration. So, study